Hello everyone, welcome back to Sowkya Vision. Today we are going to see hemoglobin. What is hemoglobin? Hemoglobin is a red pigment. As the RBC, the red blood cell, the red color to the RBC is given by red color pigment known as hemoglobin. So it is a conjugated protein. So it is a protein. It is a chromoprotein. Yes, it gives color to the cell. It containing iron part and the protein part. Is the hemoglobin containing iron part as well as the protein part? What is the normal value in the adult human? It is about 14 to 16 grams per deciliter of blood. In males, it is about 14 to 17 grams per deciliter, whereas in women, it is about 12 to 15 grams per deciliter of blood. Yes, the hemoglobin, it is a globular protein. Hemoglobin is a globular protein which helps in the transport of oxygen in our body through blood. RBC are present in blood, right? So RBC containing hemoglobin. The hemoglobin binds with the oxygen and transport in the blood. Yes. So it is a respiratory pigment. Yes, it is a respiratory pigment which has ability to transport oxygen by forming oxyhemoglobin. By forming what? Oxyhemoglobin, it transport oxygen and also transport carbon dioxide by binding with the carbon dioxide and carbaminohemoglobin, forming carbaminohemoglobin. Yes, now let's see the structure of the hemoglobin. Hemoglobin containing two main parts, globin part and heme. Hemoglobin means breakdown into heme and globin part. Heme containing iron. The heme is containing iron component, whereas globulin, it is a protein part. Globulin is a protein, heme containing iron. So this is the structure of the hemoglobin. As we know, heme is a pigment part containing porphyrin. Containing porphyrins of what? 4 pyrrole rings. Yes, 4 pyrrole rings. And now these pyrrole rings, 4 pyrrole rings are attached to methane bridges. Yes, these are attached to methane bridges. And now the iron part. The iron is attached to N of each pyrrole ring. Yes, here it is the iron, right? The iron is attached to N. Here N, here N, here N. So, it is attached to N of each pyrrole ring and N of globulin. So, let's see how globin is present. Globin is a protein part of the hemoglobin containing 4 polypeptide chains, in which 2 are beta chains and 2 are gamma chains. The beta chains are these, whereas these are the gamma chains. Yes, 2 beta chains and 2 gamma chains, 2 alpha chains, 2 beta chains and the 2 alpha chains. Now, there are 2 types of hemoglobin in the human, the adult hemoglobin and fetal hemoglobin. The fetal hemoglobin has more affinity for oxygen. Yes, it has more affinity to bind with the oxygen and transport in the blood. So let's see what are the functions of the hemoglobin. There are 2 main functions of the hemoglobin. What are they? The transport of respiratory gases and the buffer action, acid-base balance. Yes, buffer action means acid-base balance. Now, transport of respiratory gases. How it transports? By binding with the oxygen and carbon dioxide, forming oxyhemoglobin, which is unstable, and carb hemoglobin, which is also unstable. Whereas the hemoglobin has 20 times more affinity for carbon dioxide than compared to the oxygen. Yes, now let's see synthesis of hemoglobin. Synthesis of hemoglobin. The hemoglobin starts synthesized in the pro-erythroblastic stage of erythropoiesis, but it appears in the intermediate normoblast stage. How many stages are there in the erythropoiesis? Yes, there are 5 to 6 steps in the erythropoiesis, whereas the hemoglobin starts maturation, producing, synthesizing in pro-erythroblastic stage, but it starts appear in the intermediate normoblastic stage. Yes, now let's see the synthesis of hemoglobin. Yes, let's imagine this is the RBC. This whole process is occurring in the RBC. So here we go. RBC, the cell containing mitochondria as well as the cytoplasm. So here the succinyl coenzyme along with the glycine is converted into ALA by ALA synthase. So it is on further is converted into coproporphyrinogen 3. Coproporphyrinogen 3. On oxidation, the oxidase, in the presence of oxidase, the coproporphyrinogen is converted into protoporphyrinogen. It is protoporphyrinogen 9. And now it is again converted into protoporphyrin 9 in the presence of oxidase. Yes, the protoporphyrin 9 is converted into heme. Yes, is converted into heme by ferrochelatase iron. Yes, by ferrochelatase and iron. This is one and this is the second one. By the presence of these enzyme, the protoporphyrin 9 is converted into heme. Now, here the ALA is shifted to cytoplasm. See, this complete process, the production of heme part is completely done in the mitochondria. Now let's see what happens in the cytoplasm. This ALA is traveled to cytoplasm. There it is converted into porphobilinogen in the presence of dehydratase. In the presence of dehydratase, the two molecules of ALA is converted into porphobilinogen. Now, in the presence of synthase, this porphobilinogen is converted into uroporphobilinogen 1. On other co-synthase, it is converted into uroporphobilinogen. This is uroporphobilinogen. This is also uroporphobilinogen 2. Yes, the decarboxylase, in the presence of this enzyme, it is converted into coproporphyrinogen 3. Coproporphyrinogen 3. And it is by here, the heme is transferred to cytoplasm and the globin formed. These together forms hemoglobin. Yes, here the coproporphyrinogen is again going back to the mitochondria to synthesize heme. Yes, and this is the complete discussion of hemoglobin. If you have any doubt, please comment in the comment section. Support the channel, like the video, subscribe the channel. Namaste.